All right. So we've gotten to here. We've described how we can do partition delta score regression. Now we'll get away from the math for a second and talk about some actual results of what this looks like in practice. So kind of the question here is we've defined a method that lets us look at this partitioning to different categories and their respective effects on sample size. So we might want to think about the many things that we could look at. So at least to date, kind of the things we've looked at are things like functional genomic categories. So splitting out the coding regions, specific regulatory elements, these DHS hypersensitivity sites, evolutionarily conserved regions. But basically, any of these annotations that exist out of the more, cell biology, genetics knowledge about different annotations of the genome. This is certainly a space that moves quickly, so you can think about, anytime somebody discovers something new about, some annotation of some sort of functional piece of the genome, you might want to add it to the things you look at. You can also start looking at things that are related to tissue or cell type that you can get at through things like studies of gene expression, so things like GTEx. You can annotate things that are expressed in some particular cell type, or using these histone chromatin marks as far as kind of a marker of expression in some particular tissue. And so the table on the right here is just the set of functional things that went into the first paper on partition delta square, and you can see that a lot of this basically boils down to looking at what exists in the literature as far as ways people have thought about looking at different sections of the genome as having different functions, and then kind of pulling those back over as annotations that you can use to try and understand more about your GWAS. The other thing you can think about is building buffers around these annotations. So given that we've defined our annotations to be essentially a 0-1 indicator of whether or not you're in a category, you can think about being right next to a category might be important. So if we take something like the annotation that is the coding region, you have literally the set of base pairs that code for whatever gene. But you can also think about, like, if you're right next to, like, you're the next SNP over from where the coding is, you can imagine that changing that base pair could do something that gets in the way of coding. Or even maybe if you're just a little ways down and you have a mutation that makes it look like coding should start there or end there as opposed to where it normally ends, you can imagine things like those being problematic. And so we can imagine specifying additional annotations that take one of these annotations and define that annotation plus some buffer region around it that will be kind of its own annotation, so we can look at, like, is there a lower but still somewhat enriched amount of signal in those regions as opposed to actually within the body of the annotation. So in practice, if you take the default annotations from that were put together for the partition heritability paper, for the functional annotations, you get these annotations, plus for each of these an annotation that is that region plus 500 kb in either direction. Here we go. So the other question is, how do we want to interpret the results out of looking at any given annotation? So if we think about a single annotation, we kind of already defined that we can look at this heritability per SNP for that annotation. That is our tau coefficient that we've estimated and the number of SNPs that are in this annotation. And then we can define enrichment for that annotation as the kind of heritability per SNP within the annotation compared to our kind of genome-wide average heritability per SNP. So we do the unpartitioned heritability, plug this in, do the partitioned heritability, get this, and comparison gives you enrichment for some particular annotation, kind of in a marginal sense. Or we can look at testing just the individual taus that give us kind of conditional on our full multiple regression, whether or not that particular category, conditional on all the other categories that we've included in our regression with our annotations, matter. So in particular, this might be of interest when we're looking at the buffer region annotations or when we're looking at highly overlapping annotations. So you can imagine that a lot of the coding regions are conserved. A lot of the annotations that relate to tissue and cell type are, of course, going to overlap with the genes that they're actually saying are expressed in some tissue or cell type. So kind of conditioning on, so if you're going to look at, say, brain-expressed things, you want to kind of condition on the amount of general enrichment in coding things before you start looking at whether the things that code in the brain are important.
I'm a little confused. Is this just a difference in talking about like what the denominator is? You're talking about enrichment within a category, or I just lost you for a moment.
Right. So it's slightly nuanced because it actually goes back to, well, there's weird math in terms of how it actually gets implemented. But in terms of the thought experiment, you can imagine that you do the single regression. You do a regression for your single annotation and get a tau out of that, and then you're looking at enrichment. You're not conditioning on having done a multiple regression with all the other categories in the regression. Or in the case that you've defined disjoint categories, you can do the single regression and do this, and it works. Or you can look at the enrichment kind of conditionally as whether that coefficient is significant within the full multiple regression model. Does that answer your question?
Yes.
Okay. Cool. So we'll breeze through results from the first paper and a couple follow-ups. So this is looking for a set of traits that had well-powered GWAS at the time this paper came out. And we see that by and large across many traits, we do indeed see enrichment of signal encoding regions. For most things, in particular for more highly polygenic things, we see even stronger enrichment out of conserved regions. I'm not sure how legible this is, but for reference, coding is roughly 1.5% of SNPs. Conserved is roughly 2.5% of SNPs. So this is large fold enrichment, but in terms of the total heritability explained, it's still, there's still plenty of things that aren't in these regions. This is just that there's more concentrated in these regions. Much weaker enrichment in, say, various histone marks. There's annotation of regions of the genome that are quote-unquote repressed, so don't have a clear function, at least that we know of, as of some point in time for annotating these things. And by and large, these do seem to have lower than genome-wide average signal per SNP, so kind of consistent with the fact that if you have regions that have above average enrichment, you have to have some that are depleted. That's probably a decent tag where they are. And then you also get you can get down to the level of specific enhancer marks, which in the case of phantom 5 enhancers were proposed to have some outsized level of importance to immune things, and indeed it's kind of off the charts in this annotation. Yeah. Yeah, this is where we get to the limits of my knowledge about the specific annotations. Yeah, there's not much more I can say about them other than it's a specific class of enhancers that I don't know if any of the handful of geneticists in the room have a better answer. Yeah, I think MHC may fall into that annotation. Yeah.
Just tie into in the first part of, so we clearly are seeing across different phenotypes, you're seeing enrichment for specific categories.
Mm-hmm.
And when we construct the LD blocks and use them in heritability estimates, we are assuming that the chi-squares are divided naive to what sort of SNPs lie within those LD blocks. So if, for example, an LD block that's X long but has a coding region, and an LD block that's X long but doesn't have a coding region in it. So doesn't it sort of, this very extension of LDSE, doesn't it sort of make the previous assumption of LDSE to sort of calculate heritability slightly difficult?
Yeah, so the fact that we see this kind of enrichment in various places does say that the assumptions we're making for the univariate heritability are bad. Is kind of implicit here. There's a nice feature of these results, though, is that kind of the concerns about other violations in the model that aren't specific to that we, at least here we're to the point that we're conditional on a category that we're not correlated and that the variance is consistent. As opposed to genome-wide, we're not correlated and variance is consistent. In looking at enrichment, if we've got other kind of misspecification, that misspecification lives in both the numerator and denominator of the comparison. So it's both in this partitioned heritability and in the overall heritability. So we at least have some protection in terms of looking at these results against that. But you're exactly right that the results here directly imply a degree of model misspecification in the heritability estimates, which is why we are generally cautious about, how to interpret those estimates of H-square SNP.
Can you perhaps separate out LD blocks based on coding regions, or like that sort of SNPs and conserved regions perhaps, and treat them slightly separately, and then run the LD score regression? Assuming that you would expect to see slightly higher chi-square statistics for those.
Yeah, so you could do that. I mean, functionally, that's what you're doing here, right? So you're running a regression that allows you to specify separately that there's a term that's, assuming you include an annotation that is the whole genome, which is kind of the de facto standard here. So you've got the term that's the whole genome, and then you've got an extra term that allows for whatever the difference is between the whole genome and, say, your coding SNPs. That answer your question? Okay, yeah, I felt like I missed a piece, but we can circle back on that. Yeah.
Would you expect these results to correspond to like a magma analysis of a gene set that way? You use the same data sets, or using like enrich or something? Would you expect high heritability to correspond to those pathways being significant in a pathway analysis?
Yeah, probably. We'll get a couple slides further in that we'll talk about things that narrow this. So, something like an enrichment and coding region doesn't tell you much about what you'd expect out of a magma analysis, given that that's taking all genes. But we'll get to things, and I think, at least in a qualitative sense, there does seem to be consistency of the kind of domains of things we see out of a partition heritability analysis and the domains of things you see out of a magma analysis or depict analysis or the kind of annotation space analyses. So that's functional enrichment. Here's based on chromatin marks enrichment for specific tissues. So this is kind of agglomerated across a large number of samples and clustered into these tissue sets that are here. And so by and large, we find things that track with what might be our expectations about these different traits. So say schizophrenia and bipolar disorder are enriched out of things that have these expression tags that correspond to central nervous systems, tissues and cell types. Height corresponds fairly strongly to things that are reflective of connective and bone tissues. Very strong signal for immune system things for rheumatoid arthritis and Crohn's disease and so on and so forth. You start getting interesting things though, that like BMI appears to be enriched primarily as a central nervous, at least in regions that correspond to expression in central nervous system, rather than like metabolic or more adrenal processes. And there's kind of an interesting mix of suggestive things for age of menarche, though it's not well powered enough here to say much about it. But you can continue refining this, and this has been this is on bioRxiv, but continued effort as kind of the biological work gives us better annotations to start looking at narrow subsets. So looking at specific sets of brain tissues, so narrowing to cortex versus cerebellum versus striatum, and kind of different tissue samples from this. I believe this panel is based on GTEx. So you can see that for something like migraine, where we expect it to be more circulation things in the brain, you see expression in these kind of lower level striatum tissue expression related things. Whereas for more higher order thought process things, you see enrichment in cortex for schizophrenia and depressive symptoms. And there's this kind of interesting enrichment in cerebellum for something like BMI and years of education. And kind of the working hypothesis there is that if you compare the kind of cell type composition of the different areas of the brain, cerebellum is actually more strongly enriched for neurons than cortex is. And so you can show there's enrichment specific to neurons rather than other support cells in the brain. And then you can even actually start getting down to specific subtypes of neurons. So we're well powered at this point enough out of the psych-encode project, comparing expression in GABAergic neurons versus glutamatergic neurons to see that perhaps, at least for bipolar, there's a stronger role, at least in terms of proximal things that you annotate via this kind of partition heritability for things that are related to these GABAergic neurons.
Briefly tell us about these two types of neurons. What?
Yeah, so it's roughly in the same vein as excitatory versus inhibitory neurons. It has to do with which molecule, which neurotransmitter molecules they respond to.
Which one is excitatory and which one is inhibitory?
I knew that was going to be the question, and I can never remember which is which. Yeah, James looks, okay. There we go.
James.
First, it's the neurotransmitter that it secretes, not the one that it accepts. A given neuron might accept both glutamate
There we go.
and GABA as neurotransmitters. It's glutamate that's excitatory. GABA is usually inhibitory, but that can depend on like the gradient of GABA in that part or in that point in the development.
Cool. Yeah, and this kind of highlights where you reach the point that you want to talk to neuroscientists or wet lab geneticists or somebody who can give you more information about what things you want to look at in terms of annotations for your particular trait. And the dangers of talking about other people's work on your slides. A few final notes on the partitioning here thing. So code and pre-computed LD scores for all the baseline annotations are available on GitHub. I think this is being covered as part of problem set 3. In practice, actually getting estimates that have standard errors small enough to make much use of tend to rely on having very large sample sizes. You can see even on this previous slide that we're not well powered enough to say much about years of education in specific neuron types yet, and we're only seeing a couple tissue types worth of information there. So, the demands of power are high and become increasingly high as you want to get to narrow and more specific annotations. The initial recommendation was to avoid annotations that are less than 1% of the genome. As you drive sample size up, you can probably get lower than that, but it is fairly demanding. I made this point previously, but because we're talking about enrichment relative to total H-square SNP estimates, also LD score, there's at least some nominal protection against other forms of model misspecification. In particular, questions about effective sample size, which goes back to we're assuming a kind of dumb basic GWAS, not a fancier mixed model or some other analysis as input. So things like the partition heritability will tend to behave a little better even if you've done the wrong thing in terms of input than just the raw H-square SNP estimate will. And there's this paper on bioRxiv showing that we can imagine extending this to continuous annotation. So everything I've just shown you, again, is a 0-1 indicator of being inside or not in a specific annotation. We can imagine that instead of being additive in those kind of categories of annotation, that we're additive in some constant value times an annotation. So it could, this works both in terms of probabilistic assignment to an annotation and in terms of more continuous valued things that you might want to model. So kind of the nice figure here, displayed in terms of quintiles, but we can think of in terms of evaluating on a continuous basis. We can think about breakdowns of heritability with respect to, say, allele age, or some measures of selection, or indeed looking at a more fine-grained functional form for our relationship with allele frequency in terms of where the heritability is. And at least for many traits, we see trends along these lines that newer alleles that have had less time to be acted on by selection tend to have more effects. Alleles that are in regions that look like they're affected by more selection tend to have larger effects, and other kind of things along those lines here. So further extensions possible there, but should switch over to genetic correlation. So genetic correlation. Thankfully, this is faster to get through once we've done all the hard work to explain how LD score in general works. And this is the third paper here. This will advance. There we go. So everything I talked about so far was in a single trait, but we have GWASs of many different traits, and we know these traits are correlated. And there's been lots of discussion as far as various ways as far as looking at what this correlation means in a genetic context, and this kind of broad idea of overlap. So kind of the simplistic observation is that if you take the GWAS of a couple different traits that are correlated, you will sometimes see significant loci that are in the same general region as each other. There were some early efforts to kind of meta-analyze related traits. So this is from an analysis by the PGC in 2013, looking across psychiatric disorders and trying to identify variants that look like they were associated with multiple psychiatric diseases, and showing at least some things that look like they might be more generally having an impact. And so there's kind of this loose idea of overlap. But it'd be nice to make more quantitative statements about the degree of genetic overlap. In particular because it gets us a couple of things. One is that so for psychiatric things, that epidemiological literature in particular, there's a lot of epidemiological observations as far as things that are comorbid, diseases that co-occur together, diseases that co-occur with, some quantitative measure. And there's all sorts of challenges to actually trying to even begin to think about causality in those contexts. I don't think I have to tell you guys that. But so being able to at least start thinking about what proportion of that, thinking about whether the things that are correlated in an epidemiological or population context are correlated in terms of their genetic factors is a useful step. And it also, being able to do something along these lines from summary statistics and in a more genome-wide fashion than needing to look for individual loci that overlap, kind of gets us around some of the data collection challenges of needing to wait until we have a GWAS of 500,000 people on every trait to talk about what things are shared, or needing to collect the same trait. To collect all of our traits of interest in the same set of individuals, which we'll get to some results that look at genetic correlation for things where just functionally there's no chance of actually collecting data on those traits together. So you can start looking at these things if you can get at genetic correlation via summary statistics. So the challenge up front here is to define some concept of genetic correlation. So what I'm going to argue for is that we can define genetic correlation in the sense of saying that we've built our best genetic predictor for each trait based on whatever universe of standardized SNPs we're interested in. We'll probably assume this is common SNPs. And this goes back to the same kind of sum of effects for these SNPs across whatever set of SNPs you defined and some residual term. As we already talked about previously, this reflects their causal effects in kind of the flip the allele at conception sense, plus effects that they tag for genetic variants that are not in whatever linear predictor we're building. So we're going to talk about correlation of all the effects of all SNPs, where all is some universe that we define in kind of the same way that existed in the previous slide in terms of what we're going to sum across in terms of LD is where we'll end up. But so kind of given that idea, we're going to say we've done this for our two traits. And then we can think about three possible correlations that exist of this model of things, these parameters in the population. So the first thing we could look at is the correlation of these pseudo-causal effect sizes. So it's the flip the allele plus whatever you tag that's not part of the set of things you've summed. So just the simple correlation of these standardized effect sizes. You could also look at the correlation of the risk. So take the full score. So this is like your idealized population level, idealized, like, you got the tablets from the mountain, you went and talked to the oracle, you sat with the Buddha and received enlightenment about what the true effect sizes are to generate your polygenic risk score, and you've generated that polygenic risk score. What is the correlation of those values? And so these two values are distinct in the sense that the correlation here depends on that, at least amongst the things that you've measured, you're talking about only the correlation of their respective effects. Whereas here you get a correlation that says something more about if I told you your risk for, if I told you your polygenic score for educational attainment, what does that tell me about your polygenic score for income or voting behavior, or pick your trait of interest. So there's nuance and difference of those things, in that this kind of captures some sense of pleiotropy of things that kind of co-occur due to being in the same region rather than being, at least amongst the set of things that we're summing across, having the same quote-unquote causal effect. And then kind of the other thing we can think about is just correlation of our idealized GWAS betas. So take the marginal effects in one GWAS and the marginal effects in another, and the correlation of those, which is more akin to the idea that we're looking at in a qualitative sense when we look at do the GWAS loci overlap. And so if we look through the equations here, the key distinction is how we're treating this, the LD matrix in the middle here. This omega that comes out of the LD matrix of the SNPs.
Sorry, can you explain the third type of correlation?
Yeah, so this is if you had the infinite population GWAS of each of your traits and had the betas from that, and took that vector of betas for each trait and correlated those. And so you're capturing the kind of, you're even more so than you do here, kind of relative to the equations. Even more so than you do here, you're emphasizing the amount of LD that's captured in both places.
Explain the first two again.
Yeah. I imagine this is where most of the time would go. Yes, so here we're talking about, so in the clean experiment where we've managed to construct this equation for every possible genetic variant, so there's no genetics left here. We've done all the SNPs, we've done all the indels, we've done all the rare things, we've done the perfect genetic predictor of every possible thing.
It's like an OLS where we've included every single variant in
It's
the...
like whole genome sequencing.
Well, yeah, not just whole genome sequencing, but that in the regression itself, we've included every variant. So in the GWAS, we only do one variant at a time. And so that means that those regressions are going to capture also the effect of correlated variants. But these betas here are the betas from a regression where you regressed everything all at once.
Yeah. So it's like the additive component of your twin model that you've actually managed to capture all of it. So in that case, each of these vectors of effect sizes are the causal effect size, change the variant at conception, what impact does it have effect size. And so then you're talking about the correlation of those effect sizes. So not only do you need effects that are in the same general area, you actually need effects that are the causal effect of the same variant. And so, we may expect that most of the things in each of those vectors is 0, but for the things that are, so to have correlation, you're talking about things that have effects at the same place and tend to have effects in the same direction. And that's the correlation we're talking about. The distinction once we move to this kind of correlation to polygenic risk thing is that you don't necessarily need to have a causal effect of the same exact variant on both traits. But if, you can imagine, go back to our cartoon of our dummy genome, if SNP1 has an effect on trait 1, and SNP2, that is strongly in LD, has an effect on trait 2 in the causal, again, we're talking causal effects there, then the polygenic risk for the individuals will be, for the two traits will be correlated, not because there's a sharing of actual overlap of a specific causal variant, but because the causal variants live in general proximity and are correlated to the extent that the signs of these effects tend to run in the same direction or consistently in the opposite direction for negative correlation. And thus, the overall risk is correlated even though we don't have overlap of specific variants that affect both traits.
Can you just give like a made-up phylogenetic pathway example of the first one, the correlation of effects? Like when those two sort of variants would be correlated?
I'll flip back to it. So in the first slide, I had the forest plot of kind of this estimate of effect on 5 different psychiatric disorders. So we can imagine that, and there are some people who theorize that there's a general factor of mental health. So that there's some component of the genetics of psychiatric disorders that is entirely shared and is just like some general mental stability thing. And that each of these specific disorders represent that plus, things that are specific to the disorder. So you get, you've got bad mental health and you've got the piece that says you're going to have delusions versus you have the piece that says you're going to be depressed versus whatever. So we can imagine that, if we're talking about those pathways, then we can say this first piece would say that we have correlation between the psychiatric disorders because there are specific variants that predispose to all psychiatric disorders. They mess up some neuron wiring thing that is bad in some non-specific way, that there are other genetic effects that, there's some other piece that is like if you hit the first exon of the gene, then it messes up depression, and you hit the second exon of the gene, you mess up for schizophrenia. Or maybe it's different genes in the same network. But so that's the comparison you're making here, is that, these are really down to the variant level of the flip the allele at conception experiment. Is there a shared effect? Whereas this tells you more about your ability, to the extent to which, because of LD patterns in the genome, there is correlation of these risk factors. Yeah.
That makes sense.
Yeah. Yeah. And then the third one, the third one mostly makes sense as the thought experiment of that we've done the infinitely powered GWAS of both of these traits and do the lookup of, just correlate the betas between those two GWASs. That's what this correlation is. Yes?
What is the difference between genetic correlation and pleiotropy?
So genetic correlation implies some flavor of pleiotropy. And so pleiotropy is a word that gets used usually for saying that there's some genetic effect that affects multiple things. And so if you have a correlation, generally pleiotropy would apply to speaking about this kind of correlation. But in particular, this is a bit stronger than pleiotropy in that we're saying not only are the effects occurring in the same place, but they also tend to be in the same direction. So you could have pleiotropy in that all the effects for your two traits are occurring at the exact same causal loci. But if their effects are effectively random in terms of which direction they go and to what extent they affect each of the two traits, being at the same location isn't sufficient to give you correlation. So saying that there's correlation is a bit stronger than saying there's pleiotropy. But once we're talking about correlation in this sense, we're talking about pleiotropy in terms of like effects of a region as opposed to causal effects of a specific variant.
So genetic correlation is a type of pleiotropy. So you could have like two traits that are very, very pleiotropic. So like let's say that you have one trait, and for every SNP that's causal for trait 1, it's also causal for trait 2. That's very, very pleiotropic. However, if the sign of the effect on trait 2 is totally random relative to the sign of the effect on trait 1, then the correlation there is going to be 0. And so correlation may be 0 even if two things are highly pleiotropic. So one's a subset of the other, I guess.
Yeah, and so this is pleiotropy this is a subset of pleiotropy at the level of individual variants. This is a subset of pleiotropy for haplotype blocks or genes or some larger unit that you tag via LD.
So I'm sorry, I may have to be done for now, completely getting this, but so I understand that you're looking at the correlation between the polygenic risk and the scores in the second one.
Mm-hmm.
And the third one, you're looking at the correlation between the betas used to generate the polygenic score. Is that right?
Assuming that we've used GWAS betas, then yes. So if you LD preded back to try and get causal betas, then you'd be looking at something more like this. But yeah, if you're using just regular GWAS betas, then correlation to the betas that go into the polygenic risk is this thing.
It might help to point out that the first and the third here are correlations between vectors. So we're imagining that we have a whole vector of effects, one for each SNP. In the first case, it's the true effect. In the third case, it's the GWAS effect that includes the LD with other SNPs. In the second case, these are scalars. So these are just like, you're taking this some, like the additive genetic factor as a whole between two people, and saying how correlated are those.
That's a good point. Thanks.
So assuming that I got the betas out of the GWAS, why would the first, second, and the third correlations differ?
So that last thing there kind of shows actually exactly how they differ. They're both going to be different from the first in that they're going to capture correlation due to this LD thing.
I get that.
But exactly how they differ, I don't think there's a real easy intuitive way.
Yeah, there's not a clear way. So, because at the limits, all these become equivalent. So if beta and alpha are perfectly correlated or have zero correlation, then all of these are 0 or 1. And it's when you have kind of in-between correlation, it's how you want to treat the LD that, it's...
I think we shouldn't stress a lot about that
Yeah,
one. No one actually does it.
Does the second one depend on like the distribution of alleles in the population you're looking at, or no?
It does, yes. So since you're hinging off of LD, so the LD matrix omega features in what that correlation is. If you change the LD structure, then you change what the effects are. And also, if you change the allele frequencies, that changes the diagonal of that matrix in terms of variance from a particular SNP. So that also changes it. Yes.
Yeah, it was just something that it's just the idea that if you look. You need non-overlapping samples for. There's kind of practical reasons. LD score gives you. There's just better ways to do it.
Yeah. And I think our challenge in describing the interpretation of this is endemic to why we're more interested in these quantities. And if we can, given that we have found ways to estimate these quantities, we can mostly ignore that this exists. It's more as far as the thought experiment, as far as thinking about what we mean when we say genetic correlation.
Yeah, the idea is that we're just to plot the GWAS coefficients against each other, and we're going to say that we did this in non-overlapping samples. The correlation of plotting these GWAS coefficients is what we're getting in 3. And so, I think the point of this is to kind of show that it's kind of related to these first two, which I think have more intuitive explanations, but it's not exactly the same thing.
It seems like it might be more relevant to predicting replicability.
If you're doing a cross phenotype of GWAS statistics or something.
Yeah, well,
That is probably true. If you want to read more text on this, there's a nice discussion of these quantities in the supplement for the second EA2 paper, October 2016, which is where a lot of these equations came from. Okay, so this is the kind of discussion we just had. So the merits of these three depend on what you want to accomplish. In practice, at least in terms of the EA results, looking at the correlation of these vectors versus the correlation, looking at those respective correlations finds very similar things. And all three are equivalent under strong assumptions, and LD score is going to make those assumptions because we're going to at least derive under the motivation of saying we're interested in this kind of wholesale correlation of polygenic risk. So again, what does genetics of trait 1 tell you about genetics of trait 2, even if they aren't sharing the exact same causal variants? But we're going to get there with an estimator that actually lives off of this not polygenic informed predictor. Yes?
So I thought in the first one, you're using the estimates when you would actually control for everything, which basically we can never do, right? And then the third one is the one that we actually do compute. So how are you now saying that you don't use the third one, you do the first one? If you don't, we cannot really estimate it.
Well, so LD score regression gives you the first one.
Yeah, so we're estimating the first one, subject to, so I tried to belabor the point that it depends a lot about what your x is in terms of this model. So conditional on having selected some set of x, which is, in practice going to be the set of variants that we've included in our LD score. Conditional on restricting to that and assuming this model, then we're going to get at an estimate of this based on the GWAS results in terms of what we're actually accomplishing with LD score, and we'll show how that works.
So in the way, like in the 2016 paper where the correlations were very similar, does that kind of at least give sort of like a intuitive like idea that the sort of the lead SNPs were picked correctly and the rest were thrown out right when constructing the scores? Or like what does that tell us about? Is that like a check for how the polygenic score was constructed? Would we expect them in real? I mean, you said with these strong assumptions they're the same. But
Actually, the assumption that we make to get there is this assumption about the betas being independent across SNPs. So this assumption that we said at the beginning, we're like, this is really a weird assumption, we don't really believe it, we're going to make it. It's under that assumption that these two estimates are the same thing. And then we see that they're approximately the same thing, which is why that's the basis of the claim. It's like, it doesn't seem to matter much.
yeah.
Cool. I'm going to power ahead here. So how LD score does this? So we've defined this model. So let's make the moment condition statements about there being a covariance matrix for our two sets of quasi-causal betas that have some covariance. That this rho G is going to be our term of interest. We're also going to allow that the residuals may be correlated, in particular because we're going to allow for the case of overlapping samples between our two GWASs, so indexed by this kind of amount of correlation of the residuals there. And then we're going to assume that all these are mean 0, and we're going to assume that, that there's no additional off-diagonal correlation between the residual and the actual estimates. So nothing too crazy there relative to the assumptions we've been making previously. Okay, so if we take this covariance of the polygenic risk as being our target and derive through, so given that we've mean-centered, we've got this kind of, our polygenic risk in each of these cases can be represented as a sum, and just by expectations we have this sum of terms. And so what we're going to assume is this assumption that the same assumption that the SNPs, that for different SNPs, the effects within trait are independent, and the effects across trait for different pairs of SNPs are independent. So that essentially we can get rid of all the we can get rid of all of these cross terms, we can get rid of all of these cross terms, and we can get rid of the terms here where j not equal k, which allows us to say that at least in by using the expectations, we're down to just these cases where it's for a single SNP. You have the effect from the two traits, and you have the variance here. Per our previous assumptions about LD, the amount of LD not being related to effect size, this parcels out, and you're down to just this summing over this expectation that we've defined as being our rho G.
I think that those two second and third set of cross terms that you have don't actually show up. There's no place where you're going to multiply beta j by beta k in that sum, right? Well, it shouldn't matter because you've just said that.
Right. It matters. That piece matters elsewhere. Yeah, that's it. It is indeed an error on the slide. But in any case, the set of assumptions, it does matter though. So yes, we're getting rid of the cross terms, and that gets us. By getting rid of the cross terms, we get the assumption that says we can say that this covariance is this term that we've defined as the covariance of the causal effect sizes. There we go. So picking up from what we've been using to derive for LD score, we have our GWAS coefficients for our two traits. But now, unlike what we're doing with heritability, where we just care about the variance of these things, we actually care about the sign, because we want to know if the sign's in the same direction or different direction between these things. So we're going to operate using these z statistics from each of these. So just take the square off, and we've got the betas over their variances. And again, this is all the same things from the univariate case. But taking those pair of z statistics, then we can talk about expectations for the product of these z statistics. So this is GWAS z-score for trait 1 times GWAS z-score for trait 2 for any given variant j, and work through the expectations here. And so given our previous assumptions about lack of cross terms, this all partials out nicely. I'll, for the sake of time, avoid talking through how the derivation works, but the math's there. And we get down to, again, this kind of regression equation where we have this term that's due to confounding, in particular cross products of the confounding term between the two traits and cross products of the error term between the two traits. If we have non-overlapping samples, then it should be impossible for these residuals to be correlated, but you could, in theory, have similar confounding between the two studies. But so all this lives independent of LD, and then you have this term that is proportional to LD that has our desired rho G in there, along with the respective sample sizes. So just as we do with the chi-squares in the previous methods, we regress the products of these z-scores that we have from the GWAS on these LD scores to get the desired values. And so kind of the final step there is this is giving us a covariance here in this rho sub G. So we want to convert that to a correlation that's actually scaled between -1 and 1. And so we just standardize by the heritability, estimated again out of the univariate heritability. So like the partitioned heritability, we're looking more at the relationship between different estimates here as opposed to needing to have a raw estimate that's perfect based on the model assumptions. Question?
Going back to what Lauren asked about Pleiotropy and genetic correlation, instead of taking the sign c-scores, if you take absolute c-scores and then run the equation, would that perhaps give you an estimate of Pleiotropy?
It's an interesting idea.
Relationships between things really weird. Like it's not clear that the product of the absolute values of the z-scores is going to scale with LD score.
Yeah, you might be able to do it with products of the chi-squares or something like that. I'd have to think about it further. It's not a terrible idea. I just don't have a concrete answer for you off the top of my head. Cool. So for the rest of the talk, when I talk about genetic correlation, we've estimated it in this way. Let's talk for a moment about the intercept. I kind of already made this point. It kind of relates to this kind of either shared residual covariance or shared confounding between the two studies relative to these two cross terms. There's a different derivation that kind of, if we live in the absence of confounding, we can talk about this more concretely in terms of proportion of overlapping samples and the overall phenotypic correlation that gets us a nice clear functional form of that in terms of the expectation for this intercept. If we have overlapping samples. If we don't have overlapping samples and we assume no confounding, then the intercept is expected to be 0. Cool. All the same things from univariate LD score apply. We weight the regression based on functional form for heteroscedasticity, assuming multivariate normal distributions as opposed to the moment conditions we've assumed thus far. Still doing standard errors by block jackknife. The interpretation is still specific to the universe of SNPs we put into computing our LD scores. So we're talking about correlation of the effects of those SNPs as opposed to other genetic variants that we didn't include in our LD score. And if we have raw genotypes, then there's a bivariate version of Gremel that we could do that's probably more powerful than doing LD score. But we're assuming we just have summary statistics. We also having to assume that both studies now have the same LD structure and that we have a good reference panel for that LD structure.
One thing about genetic correlation that LD score regression is good for, that you only can do Gremel if you have individual level data that measures both phenotypes.
Yes.
Right, and so if you don't have that, then you can't get genetic correlation. Whereas if you just have GWAS summary statistics,
I think that's not true. You could have one cohort that has one phenotype, and another cohort that has only the other phenotype.
and you can run Gremel on that.
Yes, you can still do true.
I have to think about how that would look, but okay. James tells me I'm wrong.
All right. Sure. There we go. So limitations. Same limitations as mentioned before. We need large sample sizes. We also need traits that are sufficiently heritable. Need to stop fidgeting. We need traits that are sufficiently heritable to be able to compute this ratio between the covariance and the product of the heritabilities reliably. So if you have traits that are minimally heritable, then this ratio starts getting very unstable because you have small values in the denominator. You have small values that are being estimated in an unbounded fashion. So if they're small enough that you get negative values for heritability in some of your jackknife blocks, then it's intractable to estimate. This is one of the most common tech support questions is, I got an error that says my heritability is negative. How is that possible? / why don't I get a genetic correlation? It's because you have no heritability to be correlated with anything. Perhaps more serious concern here, the inference about this genetic correlation is only as good as the phenotype or trait that was defined in these GWASs. So if you have GWASs that have done kind of strange things in their ascertainment, you can induce kind of strange genetic correlations. So you can imagine, it's easier to imagine with dichotomous phenotypes. So if you imagine that all of the GWASs of schizophrenia actually only took schizophrenics who are also bipolar, then suddenly you have genetic correlation between that risk for what is labeled as schizophrenia, but this is also actually schizophrenia plus bipolar, and a GWAS of bipolar. So thinking about the quality of the phenotype definition is often important to interpreting these correlations. And it is a very, as kind of evidenced by the different flavors of correlation we could talk about and kind of the nuanced discussion as far as how that relates to pleiotropy. As a single estimate of genetic correlation, it's kind of vague in terms of talking about the correlation between risk for things versus wanting to think about causal relationships between the genetics of different traits and kind of the nature of that pleiotropy and whether you're actually getting pleiotropy at the variant level versus the haplotype or gene level and some of those kind of questions. All right, we'll speed through some results because, we've got 15 minutes. We've got tons of time. Imagine that. Okay, so this is what was done in the first genetic correlation paper, Brendan, first author, was to look at the full correlation matrix across many pairs of SNPs. And so kind of the idea here is less about the specific results and more to think about the kind of insights that looking at genetic correlations can give you. So kind of the first thing I highlighted here was that there's a lot of genetic correlation between sets of traits that we might expect to be correlated phenotypically. So kind of this upper block here is a lot of vaguely metabolic or health outcomes in terms of BMI and smoking and coronary artery disease. You get nice strong correlations between Crohn's disease and ulcerative colitis, which are two forms of irritable bowel disease. So phenotypically fairly similar, and indeed the genetics overlap. See nice correlation of anthropometric traits. So this is height and birth size and infant head circumference. So again, things you might expect to track together in terms of the biology of these things. And signs that there was a lot of kind of cross-correlation across psychiatric disease, which has kind of been the fodder of much discussion and is kind of, continues to feed to this question about general factor of mental disorders and to the extent that that provides information. There were also things that were somewhat new, largely in things that perhaps might be unexpected or just people hadn't looked at. So one of these is this significant genetic correlation after Bonferroni correction between schizophrenia and anorexia. So this kind of falls into the category of things that you probably couldn't look at in a single population. I would challenge you to be able to design a study with 50,000 cases that have both anorexia and schizophrenia. But by being able to do it from summary statistics, you can look at things like this and start at hypothesizing about shared genetic effects in terms of executive functioning that might is kind of the leading hypothesis at the moment. There's also kind of this cryptic positive genetic correlation between autism and educational attainment that has generated a lot of continued discussion. It wasn't actually significant here, but has become significant in follow-up. I think this was based on the EA1 summary statistics at the time, so it is very strongly significant at this point, and has continued to generate discussion in that the genetics run into this direction among common variants, but if you look phenotypically at individuals with autism, or if you start accounting for the effects of rare variants, there is much lower kind of educational attainment and in general cognitive functioning in individuals with autism. So what to make of a positive correlation of the kind of common variant effects we see in GWAS is kind of fodder for interesting research. And in some cases, you can get interesting null results. So there's, yeah.
Actually, the distribution, the mean IQ among people diagnosed with autism spectrum disorder is lower, but the variance is also greater.
Yep.
So it's like the distribution is being pulled to the left, but there's what are called high functioning autism kids.
Yep.
When I was in grad school, I had a classmate who said that I had autism behind my back. Yeah, so that's what seems to be going.
Yeah, yeah. Yeah.
I think so.
Yeah, I'd say if you want to read more about that particular case of autism and educational attainment, there's a fantastic series of papers by Elise Robinson digging into kind of the spectrum of IQ in autism cases and how it relates to both common and rare genetic variation in relation to intellectual disability and lots of things. But yeah, so there's point out no results. So some of the hits in schizophrenia are on cholinergic receptors that are classically expected to be related to smoking. People with schizophrenia smoke at enormous rates. It's very hard to find schizophrenia patients who don't smoke. But in genetic correlation between schizophrenia and the measure of ever versus never smoking in the population, little, if any, evidence of an actual genetic basis to that. It's Isu's birthday tomorrow. Yeah. Say happy birthday to Isu before you leave for the weekend. Here we go. There you go. We're closing that. So yeah, so that was the matrix in the original paper. As you can imagine, this is of interest for follow-up. So this is on bioRxiv and is particular an attempt to follow up on correlation among psychiatric disorders and look at how they compare to neurological disorders. But as part of that, have also looked at how the psychiatric and neurological disorders relate to a number of more continuous population traits. So this is kind of the section of a correlation matrix that's in the off-diagonal where we have the psychiatric stuff, the neurological stuff, and the slate of more population continuous traits here, at least a first approximation. So one of the strongest results is that the kind of educational attainment stuff seems to be broadly correlated with many psychiatric outcomes. And you get kind of points of significance here, interesting things, but just kind of the idea of the slates of things you can start throwing in here. There we go. There's the results in the educational attainment paper from last year, looking at things like brain volume. So there's a nice set of GWASs from the Enigma consortium on genetics of each of these pieces of the brain and total cranial volume. And you see the cranial volume, but not specific things here. You've got neuropsychiatric disorders, you've got behavioral outcomes, anthropometric outcomes. As you can see, many different cases where you can start looking at correlations. I'm probably the wrong person to belabor any of the particular points here, but just the idea of the kind of things you can look at. So you have your one trait of interest, you go back and you compare to other things that might be relevant in terms of the genetics. A fascinating result that is in this paper by Nicola Barbin and Melinda Mills and colleagues end of last year in Nature Genetics is looking at the GWAS of reproductive behavior, so the age at which you have your first kid and the number of kids you have total. I found some great genetic results, but also some really interesting instances of genetic correlation. So correlations that relate to other reproductive things that might make sense in terms of age of menarche and PCOS and some things along those lines, but also to a range of behavioral things. So very strong positive association that being predisposed to, at least from the genetics, towards more education is also genetically correlated with waiting longer to have your first kid. It's not in this table, but we see strong relationships between both of these reproductive phenotypes and ADHD as well. You can get into, so you can imagine that there's plenty of room to take many social science traits and other things and start looking at interesting health outcomes and kind of vice versa, and just ideas as far as ways to go. I think the final thing I wanted to flag here was in terms of, given that, the single point estimate of a genetic correlation for a pair of traits is kind of maddeningly simple about wanting to think about actual mechanisms, we can think about directions we can take that. One is that genetic correlation is great for identifying proxy phenotypes. This is a point made in the educational attainment paper that educational attainment, while a kind of rough trait, given that it's easy to collect and we have this giant GWAS, might be good at providing insight into traits that are harder to get large GWAS of. In particular, there's some examples given in the paper. I'll just point you in that direction. We can then perhaps start thinking a step further about trying to pool these highly correlated phenotypes and meta-analyze them in some fashion. So back to the idea that was on the first slide as far as what the PGC did, but perhaps better opportunities for that, taking advantage of being able to estimate the actual degree of correlation via something like MTAG that Patrick's going to tell you all about the exciting possibilities there. There's also some folks that have proposed that looking at genetic correlation across sexes might be a good way to start identifying traits that have sex-specific genetic effects. Kind of the leading example where there are signs that RG is less than 1 if you do the GWAS just in males versus the GWAS just in females is waist-hip ratio. Some signs that there's some differences in terms of the genetics of that. And you can obviously start thinking about ways to follow up on finding a genetic correlation to start looking at specific instances of causation or pleiotropy. So within family designs, trying to rule out the cases of this being ascertainment artifacts. So as I said, the genetic correlation is only as good as the definition of the trait in your GWASs. This has been one of the complaints about the autism educational attainment GWAS, and thus at least ruling out that kind of ascertainment for autism has been kind of area of active work. So I'll just kind of selfishly promote this great work by Dan Weiner and Elise Robinson on trying to show that even within families, the polygenic risk seems to travel together for autism and educational attainment. But certainly potential for many ways to follow up on what is a single value for genetic correlation.
What does ascertainment artifact mean?
So this is this idea that one way you could get this, particular to the worry about educational attainment and autism, from people who just were convinced this had to be an artifact of some sort. So you can imagine that if to be in the GWAS of autism, if you did the GWAS of autism in a way where you collected your case of autism from people who were recruited to some study, they're involved in some autism clinic, or they volunteered for research in autism or something along those lines. And you've been comparing them to some population reference sample of controls who aren't necessarily ascertained and could have come from some biobank or some very loose collection of genetics in the population. You could get a bias in the sense that the people who have volunteered and shown up to be in an autism research study may have parents who are more educated and are more motivated to get their children involved in this research and want to know more about the genetics and want to further all those things. And thus your cases of autism in that GWAS are also reflect to some degree above average educational attainment compared to your more loosely collected set of population controls. And thus you have correlation between that GWAS and the GWAS of educational attainment, not because there's anything specific about autism that relates to educational attainment, but because of the kinds of people that you've identified as cases versus the kind of people you used as controls. And so the idea for this is that you take the polygenic risk for these things and go specifically to families and do this kind of within family design. You test the distortion of transmission of risk for educational attainment from the parent's polygenic risk versus the child polygenic risk to show whether or not they're over-inheriting that risk versus just being a 50-50 mixture of their parents. But yeah, those are the kind of artifacts that are a worry in terms of pooling together lots of big studies that have mixed phenotyping quality. It's more an issue for these kind of disease phenotypes versus things that you can measure in a large population sample, but is definitely the argument against how strongly you can interpret the genetic correlations for some of these things, at least without further work. Yeah.
This maybe kind of answers my question based on what you just said, but why would we, if we had all the genetic data and we had the phenotypes, I mean, in some ways it seems like it would still be better to do LD score regression, unless what you were saying. I mean, because you're getting this kind of average effective betas, it seems like it would be a more stable estimate than doing it within sample, but based on what you just said, that might not.
Sure. So I think some of the within sample, so the within sample designs allow you to get tagging of a different universe of SNPs than necessarily you get from the LD scores. So there's, to some extent, Greml starts getting you also tagging of rare variants more so than you might get of LD score, though that's kind of fuzzy. It's more, there's also just a power argument. So given that we're using a moment-based method on summary statistics and having to make a lot of generalizations to get to our estimate, you're losing information versus the information that would be present if you could actually work from the individual level data. So generally, if you take, this has actually been done. You can take the same set of individuals, do the GCTA Greml estimate versus do the LD score estimate for the exact same phenotype, exact same design of the GWAS, and you do get roughly the same values, but smaller standard errors by doing Greml. So it's more efficient because you have more information. I think James has a comment on that point.
I also believe that BGA, Ronald Flaming, who was mentioned the other day, is going to give a presentation where he will claim that you can estimate the intercept in this LD score regression framework using GCTA. So
Interesting.
I look forward to hearing about that.
Yeah. And there are also extensions to things like partitioning heritability, the analogous to LD score that can be done with GCTA as well, that I've seen proposed too.
It's possible to use LDSC to go and determine which SNPs or blocks are driving the actual genetic correlation between two traits?
Not at the moment. That's definitely an area that we've thought about, that trying to do some partitioned heritability analog in genetic correlation. Yeah, so Hillary Finucane, who's the lead author on the partitioning stuff and second author on the rest of the LD score stuff, has a student now working on precisely that question. Last I talked to her, the primary challenge is figuring out how to interpret values out of that, because once you're into the partitioning that covariance, you're starting to think about things like partial correlations between traits relative to specific categories. So getting something that's has some intuitive understanding of what you actually mean when you estimate these values is a challenge. But there's definitely room for that potential, and it's an area of active research.
Couldn't you get something by, suppose I use the one polygenic score to kind of predict the SNPs on the other, and then see which ones looked significant. So it's a little bit like an IV instead of an OLS in the GWAS. So you're saying if I only want to get the SNPs on, suppose I want to get all the SNPs on BMI that are significant when they're instrumented by intelligence, then I could kind of get then just these kind of, just doing it like IV at the GWAS level might be a good way to identify that.
Yeah, so it's definitely something we've done. At that point, you're into more of a Mendelian randomization kind of analysis, and doing Mendelian randomization with polygenic scores is an area that a lot of people are thinking about and publishing on, and I know there's strong opinions about what
These recommend some that's not causal, though, right? I mean, the problem with Mendelian randomization is you're making causal claims, but if you're doing it, but you're not trying to be causal.
So do the same thing and just don't interpret it as causal?
Yeah.
That might wash away a lot of the concerns.
Instead of OLS, you do IV in the GWAS.
So in the GWAS itself, so.
In the GWAS itself, yeah.
Okay. And,
Oh, I see.
And we're not
what-
making any strong interpretations,
And what's
just
the
like Patrick
instrument?
is saying. So suppose I want to do, suppose BMI is the variable and intelligence is the instrument. And so whatever, I'm trying to partition the part of BMI that has to do with people being intelligent and maybe taking advice and stuff. And then the part of the BMI that doesn't interact with intelligence.
So you're gonna use the.
The interpretation's a little weird. So I mean, effectively, you're taking the ratio of the GWAS. Like I think it would be equivalent to taking the ratio of the GWAS summary statistics, right? So you'd be so if you take the IV interpretation of the first reduced form divided by first stage, but in this case, it's just GWAS of IQ over the GWAS of whatever it is you're thinking
Yeah,
about.
on a snip by snip basis.
Yeah.
I
that's
mean,
what
you
the
gotta
average
think about
across-
this more
Yeah,
carefully.
yeah.
This
Yeah.
can't be.
Well,
But
it's
I think there's correlational ways of looking at the overlap snips.
But it's
yeah.
not a polygenic. I was confused when you said polygenic score because you're not talking about using a polygenic score. You're just doing snip by snip.
Yeah.
You just
Yeah, he's
yeah.
Yeah. And there's generally concern about conditioning on heritable things in a GWAS due to the potential for collider bias. But if you're careful about interpreting what your results are out.
Will you remind us of what collider is?
How to frame it in that context. So once you condition on a heritable thing that may have that relates, so it relates to both the SNPs that are in the regression and the phenotype. So Patrick walked through the kind of the idea of you can do the regression sequentially, that if you're conditioning on some covariates, if you did the regression to residualize both the trait and your genotype on those covariates, and then did the simple regression of those two residuals, you get the same result. So the concern is that if the heritable thing you're conditioning on is related to the phenotype and is related to the SNP, you explain some fraction of the SNP with that heritable covariate, and thus, when you're looking at the estimate of the SNP's effect, it's only like the fraction of the SNP that's not explained by this heritable thing, which may, need to balance out for, may not be reflective of the actual SNP's causal effect.
It's probably that you're just holding some pathways constant,
Yeah.
right? And so that makes the interpretation a little funny.
Yeah. So it's interpretably funny, but it's possible that you can find a way to construct it that gives you an interpretation you're actually interested in. We're basically out of time, but thankfully I'm at my conclusion slide. So the last thing I wanted to say is just that there's a lot of resources around LD score regression. So I've already mentioned that the code is publicly available on GitHub. There's also the wiki on that same GitHub page that has tutorials for running each of these analyses and a whole section of FAQs, both on kind of the conceptual questions and on technical support issues. There's also a user group if things aren't answered in the FAQs. That's useful both for questions about the method and for tech support issues like I can't get LD score to run. I'll admit that this mostly serves as an alternative way of emailing me, but we'll at least nominally say that there's a user group. And then the last piece of this is LD Hub, which is hosted at on the Broad website, but is actually run by Chris Zhang and colleagues at the MRC. In the UK. But this is useful for two things. One, they maintain a nice listing of all the publicly available GWAS results for traits that are sufficiently well-powered to be running genetic correlation against, which is useful just as kind of having a central clearinghouse that will list what might be available that you can go download. And it provides an online tool that if you don't want to run it yourself and don't have concerns about letting some web tool run it for you, you can run genetic correlation against these publicly available summary statistics by uploading your own set of GWAS results to run correlation against those. So just lots of resources available to actually make use of this in practice. That's all I got.